Hello, everyone, and welcome to today's broadcast. Connect across thousands of expertly curated genomics datasets using Qiagen's immunology-focused DiseaseLand database, presented by Dr. Lucy Williams, a Senior Bioinformatics scientist at Qiagen. I'm Dr. Susie Valdez of LabRoots, and I'll be your moderator for today's event. We're delighted to bring you this educational web seminar presented by LabRoots and sponsored by Qiagen. For more information about our sponsor, visit them at www.qiagen.com. Before we begin, I'd like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box that appears on the screen. Our speaker will respond to your questions via email. If you have any trouble seeing or hearing the presentation, just click on that Ask a Question box to let us know you're having a problem. This presentation is educational and thus offers continuing education credits. Just click on the Continuing Education Credits tab located on the top right corner of your presentation window and follow the process for obtaining your credits. Please join me now in welcoming our presenter, Dr. Lucy Williams. I will now turn the presentation over to her. Welcome, Dr. Williams.
So thank you. Thank you for giving me the opportunity to describe the immunology content of our DiseaseLand database. And also, I just kind of wanted to briefly introduce myself. As you said, I am a bioinformatician with Qiagen at Omicsoft in Cary, North Carolina. And a major aspect of that role in customer relations is, in terms of software development, is to really advocate for the customer. And a huge part is to address the everyday challenges that are faced in genomics, and as well as to train customers on what the software has to offer in terms of overcoming those challenges. So for today's agenda, what I'm going to do is to basically describe the LAND database and the software solution itself, the LAND's framework. And then I'll go into an overview specifically of what immunology content is in that database. And then we will a focus look at some example use cases. And then finally, we'll end with just some summary. So again, to start off, what we'll first do is we'll talk about what the database is, and then we'll talk about what the software solution or the LAND framework is, and how that's designed to enable the scientist to overcome those everyday challenges in your research. So basically what the situation is, and what we heard from the customers, is that out there exists in the public space genomic data sets. And these data sets are really underutilized in terms of their value, because to take advantage of that existing knowledge, we find that you oftentimes have to basically reinvent the wheel. And what we mean by that is the challenge is that it's tedious and it's resource intensive to download these genomic data sets, develop the analysis pipelines to be able to visualize that data, and then finally you have to have a place to store all of that data across thousands of disease-focused studies. So what if existed a database that already had all of that data pre-analyzed and loaded into it, and that was integrated with an easy-to-use software solution to allow both the informaticians as well as the bench scientists to quickly access that data? So that's essentially what the LAND framework solution addresses. And the impact then is to be able to allow scientists to empower their research quickly, identifying the high-quality data sets that are of value to them, and for the informaticians to really focus their attention on the advanced pipeline development. So let the software do the basics and enable you to get the data out and to really expand upon that data analytics. a central feature of building that data into the land is consistency, of course, standardization, as well as quality control. And so what I'm showing here is basically the pipelines that we use to actually take those genomic data sets and to build into our LANDS database. Basically, the overview is that we start with data retrieval, and that's defined by basically going to either public projects, new projects, or, for example, private data. So, for example, a customer's data or your data. And we basically, QC those data, and we download the raw data as well as the sample and project level metadata that's associated with those public projects. Then there's a two-phase curation process, both the metadata curation, which is handled by scripting out the sample and project level details, as well as hand curation of the studies into controlled vocabulary. And then there's a data processing segment where we are taking the raw files, we're extracting the signals, we're applying standard normalizations, in sample level QC, and then we're pulling that all together at the next phase where the curators or the expert level biologists are enabling the informaticians in basically stating these are the models based on these projects that need to be defined. And then what happens in the next phase is we actually take those data and we generate inference reports. And finally, there's a post-QC analysis, which happens at multiple levels, from the sample level QC, as well as where we can hand go into the projects and pull from other literature that exists to look at things like, how consistent is this expression change based on what we know from other literature. And then finally, all of those data will basically go into that LAND database. So both the omic data as well as the metadata. So then what is the database and what's the actual LAND framework and what's the solution that we're providing? So in addition to actually processing the data and processing the curated metadata, solution here again has to do with some key features related to, for example, accessing the data as well as the data footprint. So, for example, we have the ability to store high volume data, so biobank scale data, with very quick, within second level accessing all of that data. And then secondarily, the data footprint, of course, is a major problem when you're dealing with thousands upon thousands of genomic data sets. And then in the Omicsoft solution, LANDs are actually delivered via the cloud. So we're able to, from the customer side, have a very small data footprint. In addition, data integration is a key feature of the LAND framework. And what that enables you to do is to basically browse across the different projects. So, for example, if you wanted to look across multiple projects that were all under the same disease category, do that, or multiple studies, but also data integration in terms of the types of data. So oftentimes when you have a biological question, for example, you would want to look at how the expression level of a gene might correlate with copper status. And so we can integrate both across studies as well as across data types. And then finally, we have powerful interactive software solutions. So in this screenshot, what you're seeing is many different types of visualizations that you can quickly generate and interact with in the LAND framework. So, for example, heat maps, interactive volcano plots. For single-cell data, we can do clustering. We have interactive genome browsers. And then for every chart that's in the LAND, there are associated sample level details. So, for example, you can drill down into where I'm showing here on the bottom right-hand corner of the slide. You can drill down to the sample level information, which really empowers you to be able to interpret that data. So, do I believe the data that's in there? Do I want to, for example, regroup the data and build new comparisons, and et cetera? So these are basically key features of both the LAND for the database as well as the way that the database is interacting with the software, the client-based software. In addition, though, for informaticians, we do offer the ability to dump data out through API features. Okay, so I've gone over basically the framework at a high level, and so now we'll get specifically into what immunology content exists in the database. So for the most part, our immunology content is project-focused. What that means is that we've curated in studies specifically from public sources, such as GEO, SA, ArrayExpress. And these studies are grouped based on the organism that the study was performed in, so human or mouse or rat. We can broadly do other genome references as well, so cat or dog. And the idea is that the way that the individual would interact with those data is that it would be organized on whether the study was performed in human or mouse or rat. And then across those different organism-based subsets, within that, you would have different data types. So for human, you might have data types of microarray, RNA-seq, methylation-seq, or microRNA, which could all be accessed within the same LAND database. And then the only sort of exception to that would be that we can handle the single-cell RNA-seq, but in order to be able to distinguish between the bulk and the single-cell RNA-seq, we need to be able to house that single-cell data into separate LAND interfaces, and so that would be housed into, for example, the single-cell human or the single-cell mouse individual LANs, and then broken down by the type of data, whether it's UMI or non-UMI based. So when you access the immunology content, you are getting a subscription to the data content. And so if we look at the graph here, you can see that broken down by whether it's a human or a mouse study and the year, basically 2016, 2017, or 2018, we can look at the growth of the content in terms of sample number over time. And then we can see that this is broken down based on those therapeutic areas. And so, for example, we can see that, there is a major emphasis recently to almost double, for example, the therapeutic area of immunology or immunomodulators within the LAND databases. Likewise, recently from 2017 to 2018, there was a huge emphasis on building in samples under the category of psychiatry. And again, this is a data subscription, and these increases over time are built upon customer request. And then looking at the same graph, but now looking at the samples broken down by the source instead of the therapeutic area, we can see where if the sample came from skin, for example, we've basically doubled that over time, or a huge emphasis, a lot of content in the LAND is hematopoietic system, and again, it increases significantly over the years, and we're upwards of over 120,000 different samples now at 2018. And so, again, emphasizing the concept that this is a data subscription. In addition to content, we increase the types of data which are built into the LAND, generally upon customer request, dependent upon, again, what kinds of queries are our customers interested in. The primary data type has long been microarray, although we are increasing, of course, the RNA-seq, and we're building in new data types such as the microRNA samples, which are a new data type in the human disease database. And so what I'm showing here is, in these numbers, is the total numbers of projects that are in each database. and then within parentheses are the ones that were added within this year. Again, just to emphasize the concept of data subscription and the fact that these databases are increasing continually based on customer-driven requests. And so one of the data types that I showed in the previous slide is the comparisons. And so in addition to loading in the expression level, for example, based on normalized expression level, we're also having the curators enable our bioinformaticians to build those inferences or build what we call the comparisons. So for example, if we have a single project, that project may contain samples that are of normal or diseased individuals, and then we could have untreated or treated from both of those different normal or diseased individuals. And so the comparisons are based on the metadata and extracting relevant information out of that metadata, build what kinds of inferences or what kinds of questions would we want to explore, and then loading in the fold changes, for example, into land, in addition to the normalized expression values. And here's an example of actually interacting with the software to view the comparisons. So if I were, for example, interested in the disease of psoriasis, I could basically filter down to samples that were from a disease state of psoriasis, and then I could visualize all of the different inferences or comparisons that are in the land for that disease. And so, for example, here we're looking where samples were mapped to psoriasis on the y-axis, and the number of comparisons on the x-axis. And then these are color-coded based on the type of comparison that was performed. And so here you can see that in the database we have different types based on that metadata. So for example, in the case of a type 1 versus cell type 2 comparison, we may have had that in the original study they examined, for example, an inflammatory cell versus a naive cell, and that would fall into a cell type 1 versus cell type 2 comparison. Or they could have done a disease versus normal comparison to examine, for example, skin samples from a control individual and an individual with psoriasis. And then oftentimes some of the there's some high interest in these responder versus non-responder studies. And so, for example, that's what I'm showing here. We could highlight those interactively from the histogram, and we could look at the exact comparisons and view all the metadata. So we could look and we could see here that we have the ability to look at comparisons for response versus no response. And in this case, we have a time course. So we have multiple tests from the same study, and samples were taken at baseline, 2 weeks, and 4 weeks. So this is the type of data that you can visualize and see in the land, and the type of inferences that the metadata is enabling. Okay, and so now let's say we take this example and we want to highlight those comparisons. We actually browse the database. We can do that. We can basically take those comparison IDs and we can search that database interactively using these visualization tools, and we can bring back visualizations such as, for example, the side-by-side volcano plots, where now we can look at the response versus no response comparisons across the different time points. We can interactively highlight data points of interest, and we can see, so if we have a potential gene that we think is interesting at baseline, how did it respond at the 4-week time point? So now again, we're able to interactively view across the different comparisons, and really we're building off of that data integration concept. So based on those comparisons, we can follow that the metadata is the control vocabulary introduced by the curators is the essential aspect of the land, And so what I basically mean by that, for example, we were using psoriasis as the example. And so if we were interested in diseases that we might take samples from skin, this would fall into, for example, the skin and connective tissue diseases. And so what we do is we define the samples based on their disease state, such as psoriasis or dermatitis, and those would ultimately get grouped into land into broader categories, such as the skin and connective tissue, or, for example, allergy. So by building these controlled vocabularies, we are able to take those millions of samples and organize them into land in a way that's easy to browse and pick through all of those different samples based on that metadata. So that metadata is actually organized into the land and visible to the user in a table format. And so, for example, if we look and see that we have sample IDs, we can define the subject that the sample came from, that broader disease category, the disease state, and then, for example, in some cases, if in addition to the disease state, if there's a stage, for example, as I'm highlighting here in the screenshot, we can visualize what the stage is. So we have onset. Is the sample taken from an active disease stage, or is it in remission? And then there's also the concept of the tissue category. So in addition to the disease itself, we also highlight whether or not the initial study indicated a particular cell type that the samples were taken from. And then from the cell type, we can build into that based on the tissue. So, for example, if we were specifically interested in understanding lung pathology, we may want to specifically look at alveolar macrophages, and we may, may compare that to, for example, epithelial cells. That would actually end up being a cell type to cell type comparison, and we could kind of filter that down in the land based on the broader category of tissue, and even broader based on something like respiratory tract. And that would help us to define from a broader tissue category, in addition to disease, how to find the sample in the land that we're interested in. So taking that as an example, what we can see is if we were to regroup all the data in the land based on the idea of this cell type. So in this case, we're looking at case cell type. So if we were doing a case versus control study, the cell type of the case would have been reported, and we could basically filter in the land down to the specific cell type of interest. And you can see that taken from the original metadata that's been hand-curated in, we can get fairly defined. We can identify, for example, many different types of immune cells that may be of interest. And so, again, just kind of taking that into how it works into the actual software, basically that controlled vocabulary is loaded in with the actual data, and it's organized into these views. For example, here in the middle of your screenshot, you're looking at the chart view, which is interactive. And so we can define, based on the y-axis, all of the samples and the disease state that was identified from the original project. And then the color coding does the tissue category that the sample came from. And so if we wanted to specifically hone in on, for example, psoriasis, we could find out just how many psoriasis samples there are. So there's over or just under 12,000, and we could select specifically the skin samples. And again, the metadata is organized into hierarchies, so in addition to be able to browse it in the land, we can use it to filter down the database. And so that's what I'm showing here on the left, labeled by the number 2. If I wanted to specifically identify dermatology samples, I could use a therapeutic area filter to check box just the dermatology samples. And so again, we could do those filters, we could select psoriasis, and then we could interactively select from the histogram, which is what I'm showing here with my mouse. I turned the psoriasis samples from skin pink, And when I do that, I can interact with the metadata at both the sample, the project, and even the clinical details level, and we can get exportable tables about those individual samples or that project. And then another aspect of the land is the ability to build off of not just the previous studies that we're curating in, but to create new studies by creating sample sets and then running analytics in the land. And so that's what I'm showing here. So if we wanted to, we could highlight different samples in this chart, and we could say create sample set, and we could build our own comparisons interactively in the land framework. And then again, just kind of going off of this concept of all of that metadata, the other thing that you might frequently want to do is to know if a project has what data types. So from the previous screen, I could just do 2 clicks basically to answer that question. So I could go from the previous screen where the y-axis was defined by cell type or disease state, and what I could do is I could regroup now to the project name, and I could get the actual accession ID. And then I could quickly recolor the samples based on whether or not I have RNA-seq data for that sample. And then I could interactively grab just the samples that have RNA-seq as the data type that also fall into my other categories of interest. Just kind of going back to this concept of data integration and what data exists. Again, the data is organized based on whether it's human or rat, and then but within those lands, the data type are all covered, including microarray, RNA-seq, methylation array, etc., except for when we're looking at single-cell data. And those lands are separated out based on the fact that the single-cell data is bulky because we're looking at individual cells, and we need specialized views to view that data. So just to look at the DiseaseLand content, specifically from single-cell RNA-seq, we can see basically that, again, it's grouped by human or mouse, and then we have it organized based on whether it's UMI or non-UMI based. And so in this screenshot, what you can see is that we have the UMI-based samples on the left in yellow and blue, and then non-UMI-based samples in red and green on the right. And what I wanted to emphasize here is, in addition to the numbers in terms of the cell counts as well as the types of cells, the cell types that are in each land, the fact that we're covering different library strategies within the land. So we're able to, within the land, have the different platforms and comparable across those data. And so kind of focusing in on kinds of cell types we're looking at. For example, we can see that in the mouse we have a real emphasis on the hematopoietic system and those cell types, and just kind of taking the example from the mouse land. This pie chart that I'm showing on the bottom right is really emphasizing the types of cells that are coming in from the hematopoietic cell lineage. So we're looking at the monocytes, the B cells. And so we're really able to compare across the individual cell types now and to answer biology questions about, you know, what's my marker gene expression like in a particular cell type. And again, you can download that data. So the single cell lands, because of the data processing, we've already processed the data, we've controlled everything, and now you're able to quickly just download out the reports from that, and again, kind of build off of the fact that we've done early work for you, and now you can enable yourself to basically move into the questions that you want to address with this and the analysis, the advanced analysis pipelines. Okay, so now I just wanted to just get a few screenshots of example use cases for how you might use the data that's in the land. And again, just re-emphasizing the concept here is to basically empower yourself to address those important biology questions with some easy-to-use software. So as an example, let's say that I'm interested, again, in those psoriasis samples. So we can look across the database and we can define how many total samples there are for psoriasis. So there are a little under 2,000 samples from human subjects. And how many total comparisons were performed. So in this case, we have 127 comparisons. And those are broken down based on cell type comparisons, disease versus normal, or again, the responder versus non-responder, and the treatment versus control. So let's say we had a question where we wanted to know, what's differential expression signatures for psoriasis samples. And we wanted to specifically sort of hone in on maybe cell type or the different types of comparisons. So maybe a disease versus normal comparison. But in this case, for example, we could specifically look at whether, if we were interested, for example, in dendritic cells, we could filter down directly to the comparisons that looked at dendritic cells, and we could look, for example, at inflammatory dendritic cells, specifically in skin, if we wanted to select that particular comparison. But in this case, let's say that what we're interested in is we're interested in identifying differentially expressed genes that are between lesional versus non-lesional psoriatic skin samples. So in this case, what we'll do is we're filtering down all the psoriatic samples into examples where both the case and the control were from psoriatic individuals with psoriasis. And we're going to then look at sample pathology where the pathology was defined as lesional versus non-lesional. So we can quickly do that by a keyword search. We can look at a comparison and we can say what was the contrast applied. And we can essentially say for sample pathology, show me everything that exists for lesional versus non-lesional, which is what's showing on the left. So it's really a keyword search-based filter. And then basically on the chart, under the tissue category of skin, in pink, I've highlighted those comparisons that were a disease versus normal. And we can see that we've got 16 comparisons specifically from microarray studies. So we're excluding out the RNA-seq studies at this point. We're specifically looking at studies that were performed in microarray studies. And what we can immediately do once we've made the selection is on the bottom left and say Browse Selected Comparisons, and we can immediately query the database. So what that's going to do is it's going to pull up, for example, a table where we're able to see all the comparison IDs and the projects that came from those comparisons. And then we can identify that significant gene list, and we can see the genes that were significant across basically each individual comparison. So we can look at, is a gene significant in test 1 versus test 7, or we can use the significant counts column in this table to say of 16 studies, was my gene of interest significantly upregulated in all 16 studies. So this is the type of quick query that we can do to identify a list of genes where the consensus is that they are upregulated across all 16 studies, providing robust evidence that, for example, those genes may be signatures of lesional versus non-lesional psoriasis. And then from there, what we might want to do is might want to sort of explore, based on that gene signature, what other diseases may also exhibit a similar pattern. And so what we can do is we can take the database and we can query it specifically for the disease versus normal comparisons, and we can pull back the fold change information for our gene list, and we can build these interactive heat maps where we can get the the differential expression for our genes of interest, coded as upregulated or downregulated red versus green. And then on the bottom, we have the color bars, which can define what is the disease category or what was the sample source. And so here we can see from the list of genes that I found that was a consensus across the 16 different comparison sets, we can see that to some degree there is a similar gene signature between psoriasis and inflammatory bowel disease. In addition, oftentimes we might want to understand if this is a gene signature, some of these genes could potentially be used as predictive biomarkers. And we might want to know, for example, is that only the case if I had skin samples, or is it possible that this could be a predictive biomarker in a disease versus normal comparison? So, for example, a control individual versus someone with psoriasis. And could we measure these markers and identify differential expression in other tissue types? So, for example, peripheral blood. And so here, again, we've taken the same heat map from the previous screenshot, and I've applied some filters to narrow down the disease category to specifically basically the skin and connective tissue diseases, or specifically psoriasis, and I've regrouped the data according to the sample source. So now we can look at the color bars and we identify, again, that if we're looking at skin, psoriasis samples from skin in a disease versus normal comparison, that we do have the similar patterns of upregulation and downregulation across the database. But in this case, for example, if we hone in on the peripheral blood samples, we see that that pattern is not displayed. So in this case, we will not be able to use peripheral blood as a way to measure the expression changes in this case versus control comparison. We would need skin samples. And then finally, the other thing I wanted to show is the interactive nature of the interface. And so you can see that basically at the top of the heat map there is one square which is purple. And that's because in the software I can interactively click on individual squares, and I can pull up in the details pane on the bottom right end, we can see the box plots where we're actually able to see the sample level information and the specific comparisons, in this case where we are looking at that particular gene, and we can see the psoriasis versus normal control comparison and look at the individual samples and look at, for example, at the metadata information that was supplied from that comparison in the interactive table under the metadata information in the center of the screen. And then finally, kind of building off of the fact that we have the different types of data loaded into the land, we may want to take the same gene signature and we may want to ask, so how do those genes respond to different treatments? So in this case, I've applied an additional SERP into my data where now I'm looking at two different subject-level treatments for two different drugs, and I'm comparing the skin samples versus the PBMC samples, and we can see that to some degree some of these genes, which are normally upregulated in the lesional versus non-lesional comparison, are downregulated upon subject treatment. And we could identify perhaps variability in the responsiveness across the different studies and start to perhaps credential the types of biomarkers which may be predictive of responses. Okay, and then so basically what I've shown now is how we interact with the software, and we would be taking a full gene list. But also sometimes we might have identified a particular regulator within a pathway, a particular gene basically, that we really wanted to extensively credential. And so what we could do is we could specifically query the database for that particular gene. So I'm looking at this S100A9, And basically, I'm looking across the database at every single data set that exists, every single comparison, and I've reorganized those comparisons by regrouping data on the y-axis according to cell type. And so now we can see that if we were to interactively basically select from this bubble plot, which is basically showing significant interactions by showing a fold change basically on the x-axis, and then the size of these bubbles in the plot defining the significance level. We can basically interact with that, so I've highlighted the samples, or the comparisons, in the purple box, and now we can see in the details pane, we can look at basically these DC cells, and we can look at the inflammatory populations that are known to exist within the skin samples. And we can identify that this gene in this study, it was upregulated in inflammatory DCs. So again, we can build off of all the data in the land to continue to understand the results getting from our queries. So I just quickly then went through some focused use cases specifically for psoriasis. And so far, I've mostly focused on the expression data that we have in the land. Before I end, I just wanted to go through one final data type, emphasizing again the integration of the different data types. The other type of content that we actually have curated into the land is the association studies from GWAS studies. And in those lands, for example, we have over 5,000 different phenotypes, or association results related to over 5,000 different phenotypes. And so the result in that case is at the variant level, or for example, the SNP level. And each one of those variants is going to get annotated from a number of different public databases. And so, as an example, if we wanted to sort of carry over from the DiseaseLand immunology content and really look at, for example, now at GWAS studies, what sort of content exists related to immunology, we can see now that in addition to the previous different sets that I mentioned, we have over 197 different immunology-related studies, over 762 of those which involve cancer or related measures. So we're not just looking at disease, we're also looking at phenotypes. Drug and treatment responses, over 600, and specifically for autoimmune diseases, 358 different studies have been curated into the land. And for the genetics lands, it's important to point out data consistency and standardization. So in these lands, you can browse across the different studies at the same time based on the fact that we have performed allele standardization. And so you're really able to explore and kind of filter down from those association hits based on the fact that you can query across different studies at the same time. So an example of how you might use this database is, let's say I was going to continue exploring the phenotype of psoriasis. We can see that we can actually directly, again, filter based on the metadata. So we could filter and we could look at studies which looked at just the higher level of psoriasis. We could pick that out, or we could even get more specific and we could look at the association of psoriasis and, for example, Crohn's disease. And basically what you would get out is, once you do the phenotype search, you would get tables, for example, which would list individual variants, the strength of the association based on p-value and effect size, and other metadata which was pulled from the original studies. And then if that particular variant is annotated to a gene, you would also get the gene name information. So this would be another way that you could potentially credential your gene list by not just looking at different gene expression studies, but also by pulling in comparison across GWAS studies as well. And so from there, I'll finish. I'll again say thank you for allowing me the time to basically summarize the immunology content in our DiseaseLand database and explain how you can use that to enable your research. And then I just wanted to mention that if you are interested in following up with this, I'm happy to chat. You can contact anyone in our Omicsoft customer relations group by emailing through support@omicsoft.com. And if you want to look at other use cases or just basically other tools that might be in the software, you can actually go to these websites. For example, the video homes will show you other example cases taken from DiseaseLand that we've pre-recorded. And so with that, I will end.
Thank you, Dr. Williams, for that informative presentation. And we will now start the Q&A portion of our webinar, and we'll address some most commonly asked questions by our viewers. If you have any questions you'd like to ask, please do so now. Just click on that Ask a Question drop-down box located on the far left of your audio presentation and type the questions into the box that appear on the screen. Click the Send button. Our speaker will follow up with questions via email. So let's get started. Our first question, Dr. Williams, is if I'm interested in a specific disease, how can I find out if DiseaseLand covers that focused area?
Right. So the easiest way would be to basically contact Omicsoft support, and we could basically query the database and give you a report back of how many studies might cover that disease area. And then on Wikipage's websites that we've provided, we actually would put information about the diseases and the broader categories based on the coverage that's in the land. And then I guess that segues into the concept of if it doesn't exist, this is a data subscription, so we're happy to build data into the land based on customer requests. And if you already have data, we can definitely build basically the framework with your data content and basically make private lands for our customers. And then finally, once you have a subscription to the data, you can actually download the data and compare it against other public resources that might be available. So if we don't have it in the land, you could certainly download the data, analyze it yourself, and then compare it against data that you could pull out of our land as well.
Thank you for that information. And what methods are used to process the data?
Right, so we're emphasizing the fact that we're basically building the data into them continuously. So we're using normalization methods that allow us to normalize the data as it comes in and basically do project level normalization, and then basically we're comparing against standards for full normalization. And then we're applying different levels of QC, both from the processing pipelines as well as basically hand QC the data as well. So a number of different standards are applied, and again, visualizing our wiki pages will give all the details behind that.
Thank you. And Dr. Williams, this next question is a two-part question. Is there a web-based application for browser, or do I need to download software? That's the first part. And similarly, if I wanted to compare my own data against land data sets, can we do that?
Right. So this gets to basically the full Omicsoft product line and what we have to offer. So in addition to the GUI interface, which I've showed here in my screenshots, where that you're basically getting a client software that you're actually loading onto your own computer, we also do have a web-based browser interface. And then, of course, we have the programmatic interface, which I mentioned previously. So we have basically multiple different ways that you could interact with the data. And then the second question related to can I basically run these analysis plans myself, mimicking what we've done into the land and then downloading the data out of land and comparing. And that kind of gets to the Array Suite product line, which is basically paired with the land. So the land database and the tools for it are allowing you to basically, empower yourself to visualize the database, and both from bioinformaticians or biologists. But we actually have an Array Suite product, which is primarily geared towards the informatician, which would let you store, manage, and then run the analysis pipelines yourself on both the NGS and Omics data.
Dr. Williams, thank you very much for your presentation today and your important research. I'd also like to thank our sponsor, Qiagen, for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through December 2018. You'll receive an email from LabRoots letting you know that this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's event. That's all for now, and thank you for joining us. We hope to see you again soon. Bye-bye.